Well, good afternoon. I assume everyone's getting ready for the reception. We're the penultimate session, and I can tell you, no offense to the other moderators and the other speakers, but this will be the most exciting panel for today. I'm actually really excited to introduce these three gentlemen. The technologies that their companies are bringing to market are really at the leading edge of how we deal with really complex, rich data, whether it's image data from quantitative analysis of radiology images, what's happening at the organ and tissue level, to at a pathological level, what's happening to the cells, how are cells acting during a disease state, and then integrating all that information together from the genomic data to the clinical to the radiological and pathological data, making that relevant for a clinical decision, whether that's scientists developing the next version of great drugs to treat cancer to treating a patient and their disease. So because we have a limited time frame here, what I've done is asked each of the panelists to present a couple slides about their companies, and then maybe we'll have time for one or two questions and answers. So with that, let me introduce the panelists. We have Dr. Ron Korn from Imaging Endpoints. We have Todd Huffman from 3Scan, and we have Scott Jenkins from RealTime Oncology. And I'd like to ask each of you to use some slides, present the story of your companies, and give us a great introduction. So Dr. Korn? Either way.
Okay. Thank you. It's great to be here. I think I can probably see my slides from the seat, but it's probably more formal to stand up here. We're excited to be here because what I'm going to hope to show you in the next few minutes is how we are changing the image in Arizona with radiology. I'm the Chief Medical Officer and founder of Imaging Endpoints, and our mission is really to connect imaging to the cure. And the question that we posed here is what really drove the origin of our company. So what if I could tell you on a single image whether your tumors have unique biology that can be targeted for therapy? What if I can tell you whether you're going to respond to one type of therapy versus another after you get your first dose? Whether your treatment is going to be working for a long time or whether you're developing resistance? And whether you will develop cancer or any disease many, many years before it happens. And we can do all this, as I'll show you by three examples, without the need for biopsy or special imaging equipment. So this is all in standard of care imaging. So the first example is a technology that we have in our organization that looks at the texture of every single pixel in an image, just like your iPhone. And it transforms that into very high dimensional information space. And we've shown through this analysis that we can tell whether your breast cancer or whether your abnormality on mammography is cancer or not. If it's cancer, if it's ER positive or negative. And if it is ER positive, what is your risk that you're going to develop an aggressive form of that cancer that mimics the Oncotype DX or mammoprint test that now costs $3,500? So basically we get a mammogram. As you can see here, we transform it using our textual analysis. We get a bunch of data in a curve, and then we analyze the heck out of that curve to really help decide each of those three things. And we can do that without taking a biopsy. So our vision is one day you come in for your mammogram, we detect an abnormality, we figure out what it is on the spot, and we start treating you that moment. And this can be extended beyond breast cancer. In fact, it's fairly easy to show these things in breast and brain, but showing them elsewhere is really a challenge because the body is more complex with organs interacting with each other. And we've done this for renal cell. And here we've integrated genomic information that we obtain from the internet, basically, and images. We created a library that links imaging profiles with genomics, and then we take that information and we apply it like a dictionary to unknowns, and we can show with that whether or not your cancer of the kidney, as you're seeing here, is going to be the most aggressive type or the least aggressive type, and what kind of therapy you should have. It's pretty remarkable. And the final thing, because I only have 5 minutes, is wouldn't it be really nice if I can look at your scan and tell you whether you're going to develop cancer? And we have a program at HonorHealth, which I'm part of, that's called the Cancer Interception Program. So this is not about early detection. This is about cancer interception. Can we detect cancer before we can see it and give you medications to stop it from ever developing? So this is a 79-year-old person. The arrow points to a lesion in the pancreas. This is pancreas cancer. This person was having some vague abdominal pains. And the diagnosis is really not hard to see at this time. The good news is, I'll fast forward, is that this patient's doing very well because it was resectable. But we looked at this 8 years earlier, because he had a scan 8 years earlier, and using novel imaging analysis software, we asked, was the cancer present 8 years before? And if you'd roll the videotape, and I want you to look at a peak that goes down as we move our region of interest down the pancreas, you will start to see the texture of this thing change. And suddenly you see a big peak right at the body of the pancreas. Now that's important. As we go out, you don't see it again, because that's exactly where that pancreas cancer developed 8 years later. So here's just an example comparing where it was 8 years before or 8 years later. Now again, a good story for this person, because we've controlled his disease, not necessarily cured him of pancreas cancer. So we're really interested in using advanced imaging technologies to answer questions that will really transform healthcare. And as an imaging core lab and research company, we have 100 trials ongoing now in 19 countries. And we have 50 employees here in Arizona, and Arizona has been a great state for us. We are affiliated with some of the largest institutions here, and nobody gets where they're going without the help of others. And there have been some wonderful people who've given generously to get our company and these ideas off the ground, and they're listed here, as well as our Arizona partnerships. So this is what we do when we have nothing to do, and we thank you.
Great. Thank you, Ron. So as we go from the kind of larger scale, looking at organs and tissues and being able to see more detail, being able to interpret things that aren't necessarily interpretable by the naked eye, and we're quantifying these things, now we're going to zoom in to the pathologist's view and how we can take images and really reconstruct what's going on the cellular level. So Todd, if you will.
I'm Todd Huffman, the CEO of 3Scan, and we do high-throughput computational biology. And actually, I did my master's degree in computational biology at Arizona State. And I was realizing today I may actually still be a student there, because I started my PhD and then took a break to start a company, and then my advisors left ASU, and I don't think they ever kicked me out, and I don't think I ever dropped out officially. So I may still be a student at ASU, but that was 10 years ago. And so as a computational biologist, I ended up in instrumentation because computational biologists are kind of like the drunks underneath the streetlight. They can only look where someone else has made an instrument to produce data for them. And I felt that was really constraining. And one area where I felt there was a real poverty of data was at the pathology scale. So looking at phenotype at a microscopic scale. This slide is very busy. If you are a pathologist or work with one, you already know what's on there. If you aren't, you're probably not going to figure it out in the duration of this talk. But getting a piece of tissue into a microscope is a laborious task, mostly done by humans. What we've done at 3Scan is I've redesigned the pathology workflow to make the entire process automatable. So if you're a pathologist or have a histology background, what we do is we do our staining in the tissue processing step, and then we do our imaging, or sectioning and imaging simultaneously. And here's a diagram of how the instrument works. At the center where the sample is, there's a little wedge. That's the blade. I make the blade out of diamond, so it's transparent. And I have fiber optics coupled to the back of the diamond, and I shoot light through the back of the diamond. The light comes out the bevel. Then the optics of the microscope are focused on the bevel of the knife, and then I have a line scan camera that images continuously while it slices. And so this allows us to do transmission light microscopy without using glass slides. On the right is a picture of the instruments that we build, and I'll show you what the data looks like. This is a mouse kidney, and we are flying through it. This is several, a little bit jerky. This is several thousand slices through one kidney, and if this is like a 20 meg video file, the raw data on this is several hundred gigabytes. Our largest data set for a single sample is 6 terabytes. And so we can zoom into an incredible level of detail compared to something like the radiology. The radiology is great, and if you can diagnose it there, you should. So a human can't look at that much data, so what we do is we take and we project it into 3D. So this is that same kidney. We're looking at the microvasculature. Those little dots you see, those are the glomeruli of the kidney, which do the functional work. And so we can now look at microscopic structures digitally in 3D, much more similar to how radiologists look at data than a traditional pathologist. We can do more than blood vessels. This is 3D H&E, which is the most common pathology stain. I'm toggling the wireframe on and off to show it's not just the pretty pictures, but we actually process, segments, and classify the structures so that we can reconstruct and understand the underlying 3D structures in the tissue that we're looking at. So we're going to fly through the center line of this intestine. It's much more impressive when the video doesn't jerk all over the place. And so we segment, classify, and then analyze this tissue, and so then we can look at quantitatively. And so this is from a tumor. That's just something you pulled out randomly from data we're working on. This is a histogram showing the distance between blood vessels inside of a tumor, and we're looking at this around cancers in which angiogenesis plays a role, so that we can look at the distribution and changes in the vascular structures. And so my company does this primarily on the preclinical drug discovery side. So we work with pharmaceutical companies who are trying to cure diseases that have some sort of tissue scale structure. Cancer is a big one, but we're also doing projects around diabetes, stroke, cardiovascular disease. Yeah, so that's what we do. And there's my team, and that's what I've got.
Great. Thank you, Todd. So you can see from the kind of macro scale to the micro scale, there's a ton of data being generated. And one of the challenges that our industry faces as healthcare life sciences is you're all doing such fantastic work, and you're seeing some of the commercialization of new discoveries. The problem is, as more and more data is generated that's really rich, very complicated, it all has to be integrated in some way. And those who make decisions, whether it's scientists developing new drugs or physicians treating their patients, have to make the right decision for the particular patient in front of them. So Scott's going to tell you about real-time oncology and how he has some solutions for that.
Thanks, Chris. My name is Scott Jenkins, and I'm the CEO of RealTime Oncology. So in so many of the slides and so many things that we looked at earlier today, and the things that we've heard over the last 5, 10, 15, 20 years since the advent of the human genome and prior, all these things are fantastic. We're trying to understand the individual basis, the molecular basis, the anatomical basis of disease, and we're figuring out a way to treat it. The hardest part, though, of all the things we've discussed and the massive amounts of knowledge and data we're collecting in big data places, at the end of the day, I'll do a pantomime for you, right? And so the pantomime is, this is 7 minutes, and this is about 24 minutes. And so this is a GP, and this is an oncologist. So I don't see you, I don't see you, I see you, I don't see you, I don't see you. So during that time, the doc has you sitting in front of them. They have the information presented to them, and they have to figure out, what do I do? Right? I don't have the advantage of multiple silos put everywhere where there's a whole bioinformatics group over here, a molecular pathway group over here, and somebody else who's trying to figure out how to translate the information, and somebody who's doing the interpretation to see if the data really means anything. I have a patient who's sick, and I need to pick a pill, and I have to know which one right now. And that's really the fundamentals of real-time oncology. Of the pieces on this slide that make things so interesting, 75% of all oncology drugs are used off-label, which means they don't have a protocol, which means they don't have a process. They're a guess. They're guessing. One of the stats that I want you to find, Chris told me to be very controversial, try to find the number of patients who've advanced on first protocol after original cancer diagnosis. You can't find the stat anywhere. And I haven't met an oncologist yet that says this isn't true. The reason they want to catch cancer early is that they have more time to make mistakes, to get you to the right treatment, because the industry tends to find ways to guess. The advances in precision medicine bring us more and more information, but even oncologists don't know how to interpret it. And so what we wanted to be able to do is really think about what makes patients unique. What's the drivers within cancer? And then think about some of the data solutions that occur. One of the things that we find is that for the driver gene variants, it's in less than 1%, which means you're not going to find a lot of commonality as we start to move of an N of 1. As we start to find individual natures about patients, we have to get the right drug to the right patient at the right time. And so it's not a one-size-fits-all. It's not even a one-size-fits-a-group. So I tried to build a slide which is really kind of hard to understand, but this is sort of the challenge of what we've heard from the big data plays. This is why, if anyone's from IBM, plug your ears now, Watson will get better for a little bit of time, and then it's going to get a lot worse. So when we think about ways that people are applying algorithms and statistics, big data technology, they're looking for signal to noise. Well, what happens is you subcategorize individuals. As we move across the screen, as we get more and more precision definition or precise definition of an individual as themselves, as their profile, we start to move towards an N of 1. Well, as you move towards an N of 1, you can't generate signal to noise. You will not be able to find the differentiation. You have to have firm confidence based on the profiling and being able to align the drugs to treat that individual based on that profile with the best information you have at the time. And so the approach that we like to think about, the approach that we're bringing to this solution, really has to do with Bayesian logic. It's sets theory. If the correlation exists, it exists at 100%. The more you look like someone else, the more accurate the estimate or the prediction or the close case is going to be, which is how most physicians treat medicine today. That's the way they want to treat. Did it work? Why? How similar is this patient to a patient that I have? And then what is the variance that could make it work or not work? And then with that addition of clinical experience, and I could talk for a very long time about the changes of clinical landscape, about the subcategorization of disease. We talk about breast cancer 15 years ago was one disease, and now it's nearly 73 individual and discrete diseases. And so you can't look at data from 15 years ago, and data from 10 years ago, and data from 5 years ago, and try to do a correlation. That exact same problem exists among sequence data. I don't believe we should keep sequencing data for much more than 6 months. You should keep the sample and rerun it with the current technology to do comparative analysis. And so that type of driver around being able to get real-world information and make that prediction is going to be key. We know that companion diagnostics is growing. We saw earlier about mobility and data being added. Truthfully, we want to inspect what we expect. We want better diagnostics to get to the right drug with right predictability. And so that's what we're going to see, and we continue to see that in the companion diagnostics market. So the solution that we've built allows for a simplified profiling of a given individual with the information that's available. And again, it's the molecular profile. So if it's a foundation data set, you can only you just pick the driver genes or upload the VCF file. And now it's inputted for the doc. From that, a prediction model occurs based on the types of information you have to profile the patient to tell you what's working and what isn't working for patients with that type of profile around the world in real time. You don't have to wait for a case report that's 6 months old to know that this treatment is killing people, if they're ever reported at all. You can just see it's a positive result or a negative result. And the system encourages people to put information in if something isn't working to see what else could be working, because it takes that out of the equation. As an open system that works with any diagnostics, that works with user-submitted information, so referential data on the literature side, we believe that we're bringing a tool that works worldwide in real time in the cloud as the next advance in cancer care, cancer treatment, and cancer prediction. Thanks.
Thank you, Scott. And I should mention that, of course, we have a closing keynote with Dr. Matt Likens, and definitely going to be a great closer before we hit our refreshments. A couple of questions. So one question is the cost of therapies. So we know that these amazing drugs that the pharmaceutical companies are coming out with these days, such as immuno-oncology agents, they can produce absolutely remarkable responses. I mean, it is incredible. Tumors shrink away. People who were told they only have a couple months more to live are now, living past the 5-year mark in diseases like melanoma. So as you think about, some of these therapies cost hundreds of thousands of dollars for courses of treatment. How do your technologies, your solutions help solve a problem, which is, can we be more efficient with healthcare? Can we do that by creating drugs that are more effective? Can we identify the patients that are really going to respond versus not respond? So I'd like to ask each of you to kind of think about that question and provide an answer for the audience. Dr. Korn?
Well, I think imaging really is well suited for that, because everybody who has cancer or other diseases always gets an image. And we always image a lot. Sometimes we image people to death, unfortunately. And I think that what we are trying to do is we're trying to find a profile, as Scott said, of patterns of tumors that are going to respond or have a high likelihood of responding to one type of drug or not. So you can imagine, you come in for your CT scan, we do a profile, an immune response or lymphocyte profile that we've developed, and if it's positive, you should go on the therapy. If it's negative, you should consider alternative therapy, and that will really help drive the cost down. And as you follow them subsequently, if you think they should respond and they're not through imaging, then it's time to contemplate changing the treatment regimen.
Thank you. Todd?
Yeah, so right now we work primarily in the preclinical space, so I can speak with most authority there. So in pathology, the cost of getting a single slide into a microscope is quite high, somewhere between $5 and $50 depending on what you're doing. So there are lots of things that people just end up not looking at because of the cost and hassle. With ours, through all the automation, our per slice cost is well under a penny. And so we can now image much larger swaths of biology and do a more complete search through these large spaces. So I think that improves things on the drug development side. On the clinical side of this, where I see digital pathology really changing things is it allows you to actually get that digital image so that you can analyze things across lots of cases. You have in pathologists, you have variance between pathologists, and even one pathologist over the course of a workday. And so doing large-scale meta-analysis of pathology data isn't something that produces a lot of results at this stage because of that variance. And so I think that in the long term, moving to a digital space where you can do large-scale meta-analysis across lots of cases is something that's going to improve the specificity.
Great. Scott?
Great. When we think about the ability to do predictions, it's really about any type of data you have. And so if you have a simple anatomical path report, and so we've got, maybe an image report that we have an indication, a biopsy's been done, the biopsy runs through an analytical or an AP house, they come back with a positive diagnosis for cancer, and nothing else is done. You kind of don't really know what to do. And so with the tool that we've built and is working, you put that information in and you say, well, here's what we think in rank order based on what people have used or are using around the world, both indication and contraindication. You should use these drugs, you shouldn't use these drugs based on what we see. But then the ability to suggest the next level of diagnostics that said, for the type of cancer that was diagnosed, these are the most actionable gene panels that you should run, and that would have this much impact on our prediction of what drugs you should use. And so the pancreatic cancer example, right? The ability to understand that expression of HER2/neu and doing a HER2/neu panel does, when positively indicated, has great effect with a great number of patients, all off-label, with Herceptin. And so the ability to sort of work through that process that says, for the information you have, this would be the result. These are the diagnostics that are most prevalent with the type of cancer that you've seen. These are the most actionable panels that should be suggested to you. And then if those come back as the driver gene or the indication, that changes the rank order of therapeutic suggestion, and being able to then link to a doctor who's doing that specifically off-label on a patient that has a profile similar to yours, takes the money down. I mean, if Ribituxin is $220,000 a course, you don't want to be wrong. And so you'd say, well, I don't really want all these gate factors. If you not only have the information in front of you that said, you know what, with what you have, it doesn't seem to work for any patients. So prescribing for you doesn't really work. Sharing that with both the patient in a conversation and say, but these seem to work, is really what changes the game, and it changes the economics, but it also changes the outcome. Because if you put a patient on a drug that doesn't work, you're losing time. And that's the other, and then the disease changes, right? The mutation. So being able to get the most accurate information up to date so that a physician can work as they work today in a collaborative way to ask questions of people that are working in a similar case, to get that real world direct physician to physician information, changes how these drugs are going to be delivered.
Thank you. So I just returned from AACR, which is a conference that tells you about what the latest findings are in cancer research. And your 3 technologies put together just really can change the game, right? Can really provide true progress with, accelerating everyone across the board, everyone's ability to treat cancer. I want to ask about adoption. So everyone's seen the news about Theranos. It's a tough story. One of the interviews that I thought was given about how it's hard to change ingrained behavior. It's hard to change, especially in medicine, the practice of medicine when it's so standardized and you have such a regulated environment. So how do you see, what are the challenges and some of the opportunities for you to have your solution become more widely adopted so all of us can benefit from the greater, richer detail that your solutions provide in data?
Well, for us, the most important thing with imaging is to do automation and segmentation. The things that you see here really require a human element to make a decision. That has to go away. And what you really need at the end of the day is somebody to push a button and get a very simple answer. And that is what Scott was speaking to, because we can give folks reams and reams of data that they just shrug and go, well, I don't know what to do with it. So that's the second solution we have to do, is we have to come up with a common language or a common platform that says, with my data, put it into your system, and then this is what you should do. And if things don't work when you do that, then we have to go back and we have to figure out why they're not working and what we can recommend as alternatives.
So Theranos in some ways has made my job as a CEO/scientist a little bit easier, because now there's no investor pressure to be fast and loose with anything. If I just say, we should do this, we need to be rigorous, I don't get a lot of pushback. To that end, we're very moderate in our and measured in our pathway to go from preclinical through clinical trials and into clinical diagnostics. And on the adoption side, basically as we develop high throughput screens on the pharma side, then we look to see, does this have an opportunity to translate over into like an LDT or following molecules through to be a companion diagnostic.
Yeah. And the thing is, I mean, it's not just Theranos, right? And honestly, I think Theranos is kind of a victim, right? They dangled too much money. It probably kind of worked, and they dangled too much money, and they had to keep putting the bar out there to keep raising more money, to keep going, to try to prove it, whatever, and then it all caught up to them. But that's not the crime, actually. The crime is foundation, right? 66% of your value twice in 18 months. I mean, like, there's nothing that's helping molecular diagnostics on the horizon. Like, no, there just is. I mean, Illumina loses 25% on Tuesday because they missed a number around services. I mean, it's like none of that is helping molecular medicine, right? I mean, everyone should be like shivering right now because there's a chill that's moving through molecular diagnostics, and if you haven't seen it, it'll hit you within a day or two. But I know in my heart of hearts, molecular diagnostics is the winner. It's the game changer. It makes all the difference. Without Sanger sequencing, without AFI, without single molecule next gen, and then some of the new nanotube stuff, we're not going to get a reflex genomic profile, complete genome, comparative genomics between tumor normal, which is the only indication that'll get you to the right drug at the right time at the right price. So it's coming. and then the ability to make that ubiquitously available, and then take advantage of some of the network think, right? The patient and so when I did my pantomime earlier about that 24-minute window, you've just given a patient a full-time job and their family, right? You have a cancer diagnosis. You've just been given arguably a death sentence, and you have a full-time job. And so there's no one more invested than their own cancer than that person and their family. And they have to have a tool where they can get the latest information, the best information, make informed decisions, make choices, and share that and interact with their doctor. So that's why I think the patient-doctor combination is kind of the win, and nobody's doing that. Everyone's either going all patient, which is the witch hunt, hearsay, complain. No offense, but I mean we see a lot of that, right? There's no sort of doctor interaction cross things. I mean, Cancer Commons is trying hard, but even then it's a bit of a challenge. And then the doctor side, where they're trying to transform an ingrained workflow. That's just a challenge. And so I think that combination is the driver. The market will drive the adoption of this technology.
Thank you. One last quick question and answer, and then if you could all thank me for, or thank our panelists.
I'd like to thank you.
Pardon me.
Chris, can I thank you
Not me.
now?
Chris.
Thank our
I'd like to
panelists
thank you now.
for a great discussion and great presentation. But the last real quick question is, we're in a great state of Arizona, pioneer state. Things happen here. Two of you live in San Francisco, but you have roots here or companies based here. So what is it about Arizona that really allows you to grow your business? What can you tell folks here who are doing great research and starting their own companies about Arizona?
Well, I think the free spirit attitude in Arizona is great. It's a very entrepreneurial place. People call it cowboy, but I like cowboy music and stuff like that. So I think that it's the people, our universities are really stepping up to the plate, and they are really educating the next generation. And there's this ability to cooperate amongst folks in this state. Because I think we do feel like a stepchild compared to San Francisco or the East Coast. And in many ways that drives our desire to really outperform our competition.
Thank you, Ron.
Yeah, so as I mentioned earlier, I went to grad school at Arizona State, and actually my leaving the PhD was sort of the ASU is a victim of their own success. I started a company and got into the Edson initiative, and then incubated at ASU, and it was doing well, so I took a break from a PhD to run the company, which I still am. And I think Arizona is a good place to do that. As compared to San Francisco, where I now live, there's no way I could have started my company in San Francisco because it's just too expensive to be there, and it's hard to like bootstrap your way into anything when you have such pressing cash demands. And I might still be in Arizona, except my wife is from Northern California, and that's where she wanted to be.
Thanks, Todd. Scott, last word?
And full disclosure, I did apply and was accepted to ASU for my PhD program, but I went to Minnesota because that's where I was from. But I talked to somebody the other day about, I'll give you this sort of the reality, the really good stuff, and then sort of the challenge that has to happen. It has to be concerted effort. So the reality is, if you're looking at things like cancer, if you're looking at disease, you're going to find that prevalent in an elderly population. You've got a huge migratory, desirable Sunbelt movement from the Midwest and the middle to the South. The three states you need if you're going to treat, gerontology and cancer are going to be Arizona, Texas, and Florida primarily. And so Arizona is a great place to look at the patient population you're trying to get to, bring technology in. You have fantastic healthcare. You have fantastic IT, right? And so you've got, the Motorola satellite, you've got the IBM, longtime IBM group, you've got tons of computer science applications being kicked out of the universities with people that want to stay here but can't necessarily find, tremendous depth of work and being able to apply their skills. And so I think bringing opportunities to that group. And then, the office space is dirt cheap. You can run clinical trials everywhere. I mean, it's fantastic in terms of draw that you're just not going to get in San Francisco. The challenge, of course, is the fuel for the rocket is money. And if I can't go to one of these events without running into several, tens or hundreds of different VCs at every event in San Francisco, right? This is some, cocktail party thing somewhere on a boat or a roof or something else, and it's packed with venture capitalists. And they know you to know you to know you to know you. Like it's a friend of a friend of a friend, oh, I met with them with that. And it's that endorsement by the network that drives the ability to start these businesses and to go to the next round at your seed round, at your A round, at your B round, taking you to market or finding somebody to take you out for M&A. It's this network effect of the money people. And so there have to be enough high net worth individuals who live in Arizona that you could find and get somebody to rally to invest their money to change the world. And I think that's the only thing that's missing. But we had the choice to start our company in San Francisco, and I chose Arizona because we have access to everything that Chris has built and provided within the infrastructure, and I think it's the smart move. If you're going to start a company, you need to be smart as a CEO about your money and your market, and Arizona's it. So, thank you.
Great, thank you. Thank you to our panelists, and thank you for your patience.